It's my pleasure to introduce our next speaker today, Dr. Mark Brynildsen from Princeton University. Mark received his bachelor's in chemical engineering from Rutgers University and earned his PhD in chemical engineering from UCLA in 2008. And after working 2 years as a Howard Hughes Medical Institute postdoc associate at James Collins Department of Biomedical Engineering at Boston University, Mark joined the faculty at Department of Chemical and Biological Engineering at Princeton University in 2010. Currently, he holds the position of Associate Professor of Chemical and Biological Engineering at Princeton. His research has been published in journals such as Nature, Nature Biotechnology, Nature Communications, Molecular Cell, and he's been the recipient of the Howard Wentz Junior Faculty Award and an NSF Career Award. So it's my pleasure to introduce Dr. Brynildsen, who will be talking about towards next generation antibiotics, systems levels dissection of bacterial responses to phagosomal stressors. Thank you.
First of all, I'd like to thank the organizers for this honor of being able to kind of tell you about some of the work that we've done. And I always like to give credit where credit is due first. John Robinson is one of my former PhD students, which most of the work that I'm going to talk about today is from his thesis. You know, just to give some clarification at the beginning, so I kind of gave my title before actually realizing it was a 15-minute talk. So I cut a lot of stuff out. So we're going to focus on one phagosomal stressor, and I'll speak briefly about the other ones. But you know, forgive me if I run short or long. You know, we might have a longer or shorter coffee break. So just to, you know, if you're not familiar with our lab, to give you some just background on what we do. So everything is kind of bacterial focused, so it's quite different than the talk that Julie just gave, but there's actually some pretty interesting parallels between the two, and you know, you'll see the parallels a little bit later on. And Julie and I did not coordinate anything. In the first one, we're concerned about antibiotics and the reduced efficacy as a function of time due to resistance. So in one area, you know, we try and improve current antibiotics, and we look at this phenomenon called bacterial persistence. This is where there's heterogeneous tolerance to antibiotics in the absence of resistance. In this other area, we actually look at maybe identifying new anti-infectives. So, for instance, increasing the susceptibility to immune attack of bacteria. We focus a lot on nitric oxide and H2O2, hydrogen peroxide defenses of bacteria, because a lot of different bugs have been identified with mutants and in animal models to require these defenses in order to actually establish an infection. So today I'm not going to talk to you about the top. There's less modeling in this area, whereas the modeling is very integrated in terms of what we do. So I'll focus on the bottom. But if we have questions during break or lunch or something about persistence, feel free to come and talk to me. So this was a nice review from the Grinstein group at Sick Kids, and it talked about basically the different antibacterial mechanisms that are present in phagocytes. You know, these are wickedly lethal organelles, right? So you end up, you know, hitting them with nitric oxide, hydrogen peroxide. You end up hitting them with bleach, antimicrobial peptides, proteases. You have nutritional immunity where you starve them for different transition metals. And when I first, you know, started learning about these different organelles, the first thing that came to mind, you probably noticed when I was being introduced, everything that I was trained in was an engineering department, right? So I started thinking about it as this. So it's basically, you know, the phagosome would be this reactor, and then the environment is controlled by the phagocyte, and then you have this bacterium in here that basically is trying to defend itself against all these different antimicrobials. So this is, you know, my artistic attempt in order to portray it as a bioreactor, in particular kind of like a death reactor. And actually, when we do our experiments, it looks pretty much like this. We do them in bioreactors, and we kind of, you know, alter the components of the environment in order to kind of model what's happening, what's going on in the phagosomes. And you'll see, you know, this is incredibly complex, and in my opinion, we're, you know, decades away from actually being able to recapitulate exactly what's going on in here. So we took kind of a bottom-up approach. So, you know, in particular, we focused on specific molecules. As I mentioned, nitric oxide, hydrogen peroxide are two that are very important. And we wanted to basically understand how the bacteria process these stresses in order to identify ways that they can be sabotaged. And you could potentially actually develop anti-infectives based on those strategies. And I won't talk, I'll mention it. I'll talk actually a fair amount about oxygen. But as a function of time, we started with nitric oxide, then hydrogen peroxide, then we started layering in different things. So we looked at different oxygen concentrations, the span of oxygen concentrations. We also looked at what happens when the carbon source changes and when you get under carbon limitation, which can happen in phagosomes. We're looking at nitrogen and amino acid supply. We're looking at what happens at different pHs. So there's kind of entire breadth of what can actually happen in the phagosomes. So a bottom-up approach. For when we started, we wanted to basically make NO more toxic to bacteria. So that was the idea. This is a potential antivirulence therapy. It's something that can work in concert with your immune system and help fight infection. And when we thought about it, it's really whether or not nitric oxide leads to respiratory inhibition, your inability to make amino acids, damages your DNA or protein. It's basically it's a kinetic competition for how NO distributes among its reaction pathways. Right? So if you think about it, if you have 1,000 molecules of nitric oxide enter a cell, how many attack iron sulfur clusters and synthesis enzymes? How many auto-oxidize to make N2O3, which can then deaminate DNA? How many are detoxified by different agents, such as nitric oxide dioxygenase and nitric oxide reductase? So the model organism that we use E. coli. E. coli is nice because A, it's, you can make genetic mutants and do a lot of things with it in a short period of time, and B, pathogenic strains of this actually use nitric oxide reductase, nitric oxide dioxygenase to promote infection. So this is a cartoon, a very, very simplified cartoon of the nitric oxide biochemical network in E. coli. And basically, these are its defenses. So these are ones where it can take NO and make kind of innocuous compounds. And what you really want to do is you want to prevent the bug from using these in order to promote damage to, as I was mentioning, iron sulfur cluster, DNA, and proteins. And I cut out this slide to fit on time, but I've gotten questions before, like, aren't there inhibitors to these? There are, like cyanide works really well, but that's not going to be, a very good therapeutic. So it's very possible that, directly targeting these enzymes might not be an option. Whereas if you actually target, things that help the cell actually mount these defenses, that might be a more viable option. So this is, for those of you who model, I kind of put this in here. We do our experiments in bioreactors with populations of cells. At the end of my talk, I'm just going to kind of muse where this field is going and, from single cell to population and stuff like that. There are reactions that happen in the cell, reactions that happen in the media. And this isn't complicated enough. You have gas phase reactions, so you have things coming in and out of the gas phase. So this is, if you're from chemical biological engineering background, this is a really great reaction engineering problem, right? So calculating the flux of NO through all of these different pathways. And this was our first model came out in 2013. As I'm sure everyone else too, as you keep going, your models get more complicated. They take in more aspects of the physiology. So these are the parameters associated with our most simple model at the time. And it's a deterministic model, right? It's at the continuum size. It's not stochastic. And I'm sure we'll hear about modeling the stochastic processes with other speakers, as we just heard from Julie. So I wanted to, so I love this quote. So this is George Box. All models are wrong, but some are useful. If you do modeling, I've actually walked into people's offices and they have this written on their whiteboard, right, as a motivating thing. So all models, they're kind of abstractions of reality, and there are attempts to kind of cast in math what's happening in the physical world. So there are always going to be some aspects of it that are not accurate. But really, the true test is not whether or not a parameter is exactly right or whether this is the exact structure you should have for the reaction mechanism. But can you use them for something? Can you learn something about biology or the system that you weren't able to do beforehand without the model? So I wanted to give a little bit of depth about one of our projects. So I'm just going to give a vignette about one and kind of at the end talk about just briefly where else we've gone with this. And this came out a couple years ago. To give you some context, HMP is a nitric oxide dioxygenase. There's been a lot of great microbiology and biochemistry done to establish that this is the main defense system for E. coli at aerobic conditions, so high oxygen levels, like 210 micromolar oxygen. NORV is a nitric oxide reductase, and this is the main enzyme that's actually used in anaerobic conditions. And there's been a lot of biochemistry and microbiology for that. The question at the time was that oxygen can go from completely aerobic in the lungs to completely anaerobic in the colon, right? And everywhere in between. So we wanted to know at these lower oxygen levels how the baton was passed. So at some point, HMP has to pass the baton to NORV in terms of defense, right? So did this happen at higher oxygen levels, lower oxygen levels? And is there a regime where the bugs themselves are particularly sensitive or particularly robust? So maybe there are concentrations where both systems don't work well, and maybe there are concentrations of oxygen where they both work really robustly and the cells are exquisitely suited to deal with the task at hand. So what we have here is, so we bought a oxygen control glove box where we control the atmospheric oxygen, and all the oxygen levels I report are basically what would be in equilibrium with the atmosphere that we keep in the box. So we had measured data at 50 and we had measured data at 0, and we had fit our model to that, approximately 15 to 20 parameters. And we actually got very good agreement at 50 and 0 between wild type as well as 2 different mutants, delta HMP and delta NORV, in terms of the ability of the model to recapitulate the data. What you see here is the dashed line is the optimal model. The halo is the uncertainty. So when we generate these, we basically do a bunch of different random optimizations. And then once we get a number of different models that are basically equal likelihood, we then do an MCMC approach, and we walk away from our parameters. And we keep everything that's basically within 10% of the likelihood. And this is kind of the uncertainty. So, if we were to so I actually think that both regimes where you have very tight error bars and very large error bars are interesting, right? So if you have models where all of the different models agree that this should be the experimental result, that's a great test to see the accuracy of your model. But you might even learn more if you go to places where your model's like, I don't know, I don't know what's going on, right? So you can see here the uncertainty kind of increases, especially in the 0 to 10 regime. And this is another way to look at it. So this is the time to clear nitric oxide, so fall below basically a 0.2 micromolar concentration. So it's extremely tight. At 50, it's tight at 0, and then it kind of balloons out of proportion between 0 and 10. So we wanted to resolve this uncertainty. So we did experiments at 20, 10, and 5 micromolar. We did 5. I've gotten questions like, why didn't you do something over here? And it actually corresponds to the resolution of our glove box. So we have basically a plus or minus 0.5 micromolar concentration control. So if you do something at 0.25 or something like that, you're not going to actually know, right? So we eventually did go to lower concentrations, like over here, and gave, basically, we predicted lower and higher according to the resolution on the box. And I didn't put that here for time's sake, but we were able to capture it pretty well after we were done. But you'll see here, like 20 is okay, that's not bad. 10's not great, and 5's even worse, right? So model didn't do great, especially in the 0 to 10 regime. And then the other thing, which was surprising, were oscillations. So this wasn't predicted by the models. And they actually happened at both 5 and 10 micromolar oxygen. So what we wanted to do first was we wanted to figure out why we had a magnitude problem here. So this is even worse than the worst simulation, the worst prediction that we had in our ensemble. And then we also wanted to understand what the mechanism was behind these oscillations. So I usually go into a lot more detail in longer talks. So we went through like a systematic modeling experiment back and forth. It happened in 4 stages. My student was probably ready to rip his hair out around stage 3. So in stage 1, we trained the original parameters. Instead of just on 0 and 50, we trained it on all the data. We couldn't actually capture all of the data. We especially had problems at 50 when you considered all of it, and there were no oscillations at all. So we then thought that, well, it could be a parametric problem with the model or structural. So structural problems are much more hard to deal with. So what we did at stage 2 to really assess whether or not we could parametrically capture this is we released all of our cellular parameters. So all of the cellular parameters have been measured in different systems, and they were kind of approximations. So we released them by an order of magnitude, and we again could not capture everything. And we calculate, we use Akayuki information criteria to compare different models when there are different parameterizations. And Akayuki was telling us, like, you better not do this. This is a bad idea. Way too many free parameters in terms of actually the improvement in the goodness of fit. And here, basically, after this, we kind of knew we had a structural problem. And I had my student just sit down to me and start talking about the experiment, just every step, every observation. And from that, he basically conveyed that at the different oxygen concentrations, the growth rate was dropping. And we know from very pioneering work in the 50s and 60s that growth rate and translation rate are very tightly coupled. And we had no dependence of translation on basically oxygen level. So what we did was we added dependence on the oxygen level because that's what we observed in growth. And we actually were able to capture the clearance times pretty well, and it kind of started oscillating. So whenever we do structural perturbations, even parametric ones, whoa, okay. Whenever we do that, so we have experiments going in to actually show that the translation rate actually did vary with oxygen level according to our predictions. And then basically here with this wiggle, we boiled everything down to a minimal model that would oscillate. And then it was dependent on parameters from basically cytochrome parameters from one paper. And we kind of let those loose a little, and we were able to capture the oscillations. And again, so we used the model to figure out what the mechanism was. And basically, this is what the model was saying. So this is the nitric oxide dehydrogenase. It uses oxygen to deactivate NO to nitrate. NO reversibly inhibits the cytochromes, right? So what was actually happening was you had, during this period, HMP is being synthesized, which will then, so NO accumulates and inhibits the cytochromes, O2 then goes up, and HMP uses it. HMP then uses it to get rid of this, and when this gets down to a certain level, the cytochromes reactivate and kinetically outcompete HMP for oxygen. So with the model, it was telling us we should see NO go down first and then oxygen, so we should see off-phase oscillations. And that's what we observed experimentally after getting this prediction. It said that if we remove this arm, it should clear at long times. So genetically remove this arm, it should clear at long times and not oscillate. That's what we observed after we measured it. And if you get rid of the cytochromes, you should clear early and not oscillate, which also was what we saw. So we had pretty strong evidence that this was the oscillatory cycle that was active. And I'm going to try and rush through this. So there's like 800 organisms that have, it's only 2 genetic components for this like stress metabolic oscillator. Pseudomonas is very different than E. coli. It's also gram-negative, but it runs a different metabolism. It's got different kinetics, different number of cytochromes. But under low oxygen, it also oscillates. So this is kind of, I was going to go through more in depth, but I said we do a bottom-up approach. So we kind of started with NO. We did H2O2. We recently put them together. We also start layering in nutrient availability and its impact. So it's really this kind of layering on and increasing of complexity as time goes by. And we really observe interesting things that when you combine these stresses. This is the recent one where H2O2 has an inhibitory effect on nitric oxide detoxification, but NO does not inhibit H2O2. So it actually has a lot of parallels to carbon catabolite repression for consumption of nutrients. And then, so these are my lab members. John, this is John's work, funded by the NSF in Princeton. And then just as a final thing, so looking forward, when I asked Marcus just to, what should I prepare for the presentation, he's like, kind of extrapolating would be good. So I had one slide about extrapolation. So, the target just overall for the field is improved capacity to predict and control biological outcomes. And for us, we're more interested in kind of our interest area is bacterial infections. So for stepping forward, we really need to get to more realistic bacteriophagocyte encounters. So as I said, it's bottom up and we're increasing complexity, but we're far from being there. And we need to better mimic the phagosomal environments, which is layering on these different components and stressors, but we also need more precise knowledge of the phagosomal variables. So I've seen references where the concentrations are an order of magnitude different, and it's not clear, even the timing matters too. Phenotype diversity in bacteria and phagocytes. So, persistence is a phenotypic heterogeneity. It's like 2 different cell types. This also happens in phagocytes, right? So I just went to a phagocyte meeting, and they called it the phenotypic rainbow of macrophages, right? So it's not just one macrophage. There's different tissue-resident macrophages, monocyte-derived ones, and each one has a specific history to it, right? So, and then, at what point, in terms of resolution, everything for the infections here is cell-to-cell combat. But at what level, you really want to predict the population level battle, like who wins, right? The innate immune cells or the bacteria. And then there's also a lot of, calling a bacterial cell dead is not as easy as a person or animal, right? And there's predicting it is even harder. So I'm sorry I'm over time, but I thank you for your attention. I'd be happy to answer any questions.
So before we start Q&A, I'd just take one second to again ask people to move in a little bit. We still have people looking for seats. Appreciate any efforts to provide more seats on the edges. And as I said before, we have people with microphones that are willing to come to you if you have a question that you have. Please raise your hand down in front.
Hi.
So if I can summarize what I think I heard, your early models made too many assumptions with too many fixed parameters. You relax all those things and let the software run around and try to find better estimates for those parameters and then more accurate predictions of the real world.
No, so we actually, the first model didn't work. We relaxed the parameters. That didn't work. We then also, we removed all that relaxation, and we added a structural change, a very simple structural change to one reaction rate equation. And that actually allowed us to fix everything. So the parametric complexity of the final model is not too far off of actually what was done initially.
One bad assumption in the first model.
There was one, I guarantee you there are more bad assumptions, right? But again, it's like, the bar is, does the model help us learn something? And I think it did. So, Marcus.
First of all, lovely. I wonder, like in the case of H. pylori, it lives in the stomach line and creates its own like base cloud, right? So it kind of reacts to its environment by trying to counteract it. So as part of this model, you could imagine, and even in an oscillatory way, something where the bacteria is responding, it's changing the environment.
Yeah.
Right? And then that, in turn, probably is even changing things in the phagosome, right?
Yeah. So like, like I didn't get to say this, but we're going into different bugs. We're going into Pseudomonas aeruginosa, and whether or not a bacteria survives, it's whether or not it can tolerate being in the phagosome. It can escape. It can it can inhibit NADPH oxidase, iNOS. There's all of these different pathogenic strategies. Even your recent work with Salmonella, right? Like having a different response than the native E. coli that's being phagocytized. That's all very, I think, important and interesting. And we're just not there yet to incorporate that. Yeah.
Okay. Thank you so much.